Excellent. Thank you, Maria. Can we turn the volume down just a little bit? Because it sounds really loud, and I like to speak a little louder than this. I feel like I'm almost whispering. Put it here. Is that better? Yeah. Okay. We'll see if it stays. All right. Thank you. Well, I want to thank Maria for inviting me to be here. I was here, we can't figure it out, many years ago, somewhere between 6 and 10 years ago probably, and got to tour the labs and visit with Susanna. And I'm really excited to be back, see all the changes and the wonderful things that are happening here. Lots of exciting research. And Maria and I actually, I was bumped into her at ICER and just mentioned I was really excited because I was coming to Spain on vacation. And she's like, well, you're coming to Madrid and you're going to give a talk. And most people might go, oh, forget that. No, and I was like, yes, because this is exciting and I just love being able to come and see the research and meet with everybody. So thank you for hosting me, inviting me, and having me today. This is great. The work I'm going to tell you about is data that we collected while I was at the University of Rochester. I moved last August to the University of Waterloo. We've continued to analyze a lot of that data and expand on what we've done, and we're rebuilding systems right now at Waterloo that hopefully will be able to continue along this path. And so I'm going to, conscious that a lot of you are familiar with a lot of the work that's done in the eye, and our goal is really to look through the anterior optics and correct the anterior optics in order to look at the retina and look at the individual layers at the back of the eye. And we want to do this at the cellular scale so that we can understand how things change in retinal disease and how things change in retinal function, layer by layer or cell by cell. And so that's really the long-term goal, and we're working on getting to that. Multiple layers of the retina that we're going to image. I'm going to talk about several of them as we go through. But again, we have to correct the anterior optics. A lot of the imaging we're doing is using a scanning laser ophthalmoscope or scanning light ophthalmoscope. We're scanning the spot across the retina. And so really having a tight point spread function at the retina is key to getting the high resolution that we need to see cellular scale resolution. And as you all know, typical eyes are aberrated. We're going to use adaptive optics to correct those aberrations. And typically, as you all know, the wavefront aberration is corrected with the adaptive optics. And again, when you think about scanning that spot across the retina, it is really key to have that aberration correction to scan that small spot. That's the gist of my AO introduction. I won't go into any more with that. But with that technology and with that capability to scan that small corrected spot across the retina, a lot of different types of imaging have been enabled. And so, oh yes, lights out would be great, thank you. And so you can see on the left the foveal cones. Those were imaged by Alf Dubra and Yusufu Sulai. And on the right, images of the rods and cones. Of course, the cones being the larger circles, the rods the smaller circles. And we can get beautiful images of the photoreceptor layer and reflectance. As we start to look at other retinal layers, it becomes key to start adding other techniques from microscopy to inspire us as to what we can do in the living eye. And so here are just some sample images using non-confocal aperture types of imaging, be it split detection, multi-offset, various other techniques. For example, in the upper left, you see vasculature imaging in mouse of single blood cells flowing, and you actually see them stop, arrest, and then reperfuse into the vessel. On the bottom left, you see an immune response in a mouse of immune cells coming in and out. I apologize, that video seems to have stopped. But you could see them infiltrating into the tissue as well as coming back and going back into the vascular space. If you're interested in that, it's a great paper, again, out of Jesse Shalek's lab. And you can also see cone inner segments, other horizontal cells, lots of cell classes that are typically transparent have been made visible using these non-confocal techniques. Another way to look that's at the retina is using fluorescence imaging, either naturally occurring fluorophores, such as the retinal pigment epithelium mosaic, which sits behind the photoreceptors. On the left, you see an image of the fovea and the RPE mosaic there, where the fluorescence is coming from lipofuscin within the RPE cells. And that is sitting in the cytoplasm, and the dark spots we think are basically where the nucleus is blocking part of the fluorescence. It's not completely devoid of fluorescence, it's just we get less back from that region. And so you can see that beautiful honeycomb mosaic in the RPE, that's autofluorescence. You can also add fluorophores in various animal eyes, such as the image on the right is ganglion cell function, where a genetically encoded calcium indicator has been added to mouse retina. It's in the ganglion cells, And the upper right-hand corner shows you sort of when a stimulation of light is coming on and off. And what you see is when the cells are stimulated from the stimulation in the ganglions, the photoreceptor is receiving the light and the signal going through the ganglion cells. The calcium in the cells goes up, which means the fluorescence intensity goes up. And so you see cells getting bright. And if you actually look, the two cells, see if I can get the mouse working up here. I'm pointing. There we go. These two here were counter flickering, and one was going on, one was an on cell and one was an off cell. You could actually see them in counter flicker. This inspires us, and inspired my research lab, into thinking about, are there ways that we can combine measures of function and measures of autofluorescence? And so can we non-invasively understand retinal fluorescence, retinal function? What's going on in the retina naturally without adding additional fluorophores? And that was sort of the gist of what we can do and where we're heading with what we're trying to do. And the idea being things that we can do in microscopy really inspire what we can do in the lab. And so my group started looking at, see, I seems to have frozen. See, I can switch here. There we go. All right. So we wanted to tap into more fluorescence, and could we understand how the retina functions, thank you, with using fluorescence as a technique. And the reason that that's exciting is because if you think traditionally, clinically, people look at retinal fluorescence. Image on the left is from a Heidelberg system, and it's an autofluorescence image, and it's really thought to come primarily from the lipofuscin. But lipofuscin is not the only fluorophore within the retina. Within the RPE cells, you also have melanin, which fluoresces. There's fluorescence coming from the retinoids within the visual cycle, within the photoreceptors in the RPE. You can get fluorescence from macular pigment located in the fovea. Now, here in these particular images, it looks dark because the macular pigment's absorbing some of the visible light, but macular pigment itself will actually also fluoresce. You have metabolic fluorophores, NADH and FAD, involved in all cells throughout the retina, as well as a lot of structural molecules such as collagen and elastin that are also autofluorescent. So if we can tap into any or all of these and start understanding how they change in different situations, then maybe we can start understanding how the retina functions on a bigger picture, especially doing that non-invasively. And when you start to think about fluorescence, most people think about spectral properties. Here's just a host of fluorophores, and they have, of course, a whole host of different fluorescence spectra. But spectral properties for some of the molecules that we're interested in become incredibly difficult because they're overlapping. For example, the metabolic fluorophores and the retinoids tend to have very overlapping excitation and emission spectra. So spectral separation becomes very difficult. And so we start thinking about other properties of fluorescence. And a simple Jablonski diagram here, very simplified. But there's a time delay between the excitation photon and the emission photon. And that time delay between excitation and emission relates to the fluorescence lifetime, and we can use that property as a property of fluorescence that's unique to different fluorophores. Not only is it unique to different fluorophores, it can be unique to a specific fluorophore in different states or different configurations. And that's really what we're going to focus on for the remainder of the work that I'm going to show you, is our fluorescence lifetime applications. Now this works by sending in a train of pulses of fluorescence. We use time-coordinated single photon counting. When we occasionally get a photon emitted, you see that time delay between the emitted photon and the excitation photon. If you, over several iterations, measure those time delays, you can create a decay histogram of the number of photons for each time delay, and with that, that creates your sort of base parameter that you're going to then fit for each pixel. You have a histogram in your image, and you can analyze that, for example, by fitting a multi-exponential decay. And often it's simplified beyond that into, we use in our case a double exponential, and you can calculate a mean fluorescence lifetime, tau mean, and that is a weighted lifetime average. So A1 being the amount of that fluorescence lifetime, that contribution, and the tau 1, for example, being the short lifetime contribution, tau 2 being the longer lifetime contribution in the exponential. And so just as an example image, I will come back and show some of this again later, but an image of the RPE mosaic on the left in one of our people, and on the right, you could see a fluorescence lifetime image map. Very different pattern, different structure, but we can start looking at fluorescence lifetime, and this is the tau mean map. You could also, though, analyze it in Fourier space by going into a phasor plot where you take the discrete Fourier transform and evaluate it at the pulse repetition frequency of the laser that you're using. And if you then plot the sine and cosine components onto your phasor plot, you get a single point for each pixel in your image or each histogram that you fit. And here's an example then for that same image of where this might fall on the phasor space. You can see the cluster there. And each point then represents one pixel in the image. The arrow on the bottom right, that would be then short lifetimes, and over near the origin we see our longer lifetimes. Everything falling on that semicircle is a single exponential component, and you can see the values there of what the lifetime would be for those single components. Now you can see that our actual data falls not on that circle, but inside. And when that happens, it tends to be thought of that it's a combination of multiple fluorophores. And so if you had a double exponential, for example, an assumption there, it would be two components and it would fall within that line. But this could also be a combination of three components. And so it would be a triangulation there of three different components contributing if you were to do sort of triple exponential decays. You can do 4, 5, 6. It can be a combination of many different fluorophores contributing, many different components contributing to that one set of data. And so what's nice about moving into phasor space is there's no assumptions about the number of exponentials or the number of fits or the number of components that you need to consider. What we can look at is where our clusters are relative to known fluorophores, and I'll talk about that a little more detail as we move forward. So now that we've sort of introduced fluorescence lifetime, retinal imaging at the cellular scale, I'm going to give you two examples of how we've been starting to look at this to understand how the retina is functioning. And the first is in monkey using 2-photon excited fluorescence ophthalmoscopy. And so the fluorophores that we're really been interested there in the monkey have been retinol or retinoids in the visual cycle, as well as NADH and FAD. And you can see that those excite below 400 nanometers, except for the FAD. It has a slightly longer excitation. And of course, the challenge in doing that in a primate eye or a human eye is the ocular transmission does not allow us to really excite at those wavelengths. Also, a lot of additional safety concerns as you're starting to use the UV. So of course, 2-photon fluorescence, we can excite at a longer wavelength. We get equivalent excitation in the short wavelengths, and we can collect a broad emission. And we, because 2-photon excitation is very limited, it's not as efficient as single photon excitation. We use a very broad collection window in order to capture as many photons coming back out of the eye as possible. We're fairly light starved. So we capture, in this case, from about 400 to 550 nanometers with a 730 nanometer excitation. And so the system that we used in our non-human primates was built by Robin Sharma as part of his PhD thesis. It's very typical scanning light ophthalmoscope. You can see multiple sources. Here, if I can move this. All right, I'm working out the mouse here. All right, so we have our femtosecond laser coming in. We scan through the system with 2 scanners here in conjugate pupil planes, a deformable mirror for adaptive optics correction, and we reflect into the primate eye. Our primates are anesthetized and paralyzed during imaging. We have additional correction through our scanners that provides optical stabilization as well of the image as we're recording. The light is then captured directly out of the eye into a PMT. This is for our 2-photon signal. We descan the reflected fluorescence excitation beam, as well as Shack-Hartman wavefront sensor for wavefront sensing. A couple of key things, we do a lot of dual focusing to focus the wavelengths into different planes and make sure that we have everything focused at the same time. This reflectance signal here is key for registration of our general intensity images. We are not able to do, in this case for the primate work, registration of our fluorescence images for the fluorescence lifetime, but I'll talk about that and how we correct that later. And this was work that actually started with David Williams when I was a postdoc, and with Chris Balcheski and Grzina Palcheska. And together we captured the first images of monkeys and did some of the later work with Robin, and then have since moved on to separate projects in separate directions. So on the left, you see a typical reflectance image, the cones being the large circles, the rods being the small circles. You can see variation of intensity across the cones. There tends to be various intensity patterns. And on the right is our 2-photon excited fluorescence image, much more uniform, much more delineation of the cones and the rods. And this is because we're not looking at any sort of wave guiding properties or reflectance properties. You're looking at the fluorescence distributed throughout the cell, right? It's the fluorescence that's there, it's the molecules that are there. And so you can see then this very nice cone and rod pattern. We started initially by looking at fluorescence intensity variations, and Robin did a lot of work to just show inconsistency with the work of Palcheski's group, that fluorescence the stimulation of the retina leads to increases in the fluorescence, increases in the molecules that we're looking at. And our evidence suggests that this was related to an increase in the retinal within the visual cycle within the photoreceptors. Particularly, this is data from rods. We applied this to look at a model of retinal degeneration, where we could see that this response completely disappeared. And we also looked at a model of systemic hypoxia, where if we made our monkeys hypoxic, we could see a slowing of the response and the slowing of these curves. But I'm not going to talk about the intensity work. I just wanted to introduce it because it does probe the visual cycle in that way. And then 2 graduate students of mine, James Feeks and Sarah Walters, teamed up to start looking at, could we apply fluorescence lifetime to the 2-photon fluorescence? And on the left is an image, again, that fluorescence image of the rods and cones. And when we did fluorescence lifetime, we were actually really surprised to see this beautiful differentiation between the rods and cones, with the cones having a longer fluorescence lifetime. And we started to probe this in more detail and actually moved into phaser space fairly quickly with another graduate student, Kang Huynh, who found that when we moved into phaser space, not only could we separate the rods versus the cones, but we could actually separate out the rods versus the ML cones and the S cones. Each of them, this is data from 3 different monkeys, 43 locations, looking at the average of the S cones, the ML cones, and the rods in each location, and we see very clear separation. And the percentage of S cones, all the evidence we have is very suggestive that this difference is very, very well related to the S cone mosaic. And so why? Why are those S cones different? And this is where we started moving from observations of what we could see and observations of how can we differentiate different cell types to what can we understand about the retina and where are the origins of the fluorescence that we're looking at, which is really the background motivation as to why we moved to fluorescence lifetime. And so we did our AOSLO imaging, 2-photon fluorescence, in 2 monkeys and compared that to a host of fluorophores that we prepared in solution in cuvettes and measured on the same system. And that's key, is variations in the systems. You really need to do your own calibration and your own generation of what we call a phaser fingerprint. And so if we take our base phaser plot and we start adding fluorophores to it, we can see on the upper left is FAD, on the bottom right is free NADH. As we started shifting the NADH from a free state to a mostly bound state, you can see that its fluorescence lifetime shifts. This free to bound NADH difference in fluorescence lifetime has actually been used quite frequently in cancer research to look at differences in cancerous versus non-tumor cells. And so it creates sort of a spectrum there. The retinoids we looked at were all trans-retinol and all trans-retinal. They show up on different spectra here. As well, we looked at retinal palmitate. We have retinoic acid in the bottom right and elastin in the upper left. A whole host of fluorophores that are potentially in the retina that we've now characterized within. This is not by any means a comprehensive list, but this is an initial starting point of the ones that we thought would be the potentially the biggest players in our fluorescence signals. The free to bound NADH, what's often done is you create a line, and it sort of becomes a line that you shift across. If I use my mouse here, you can shift across this line as the ratio of free to bound shifts. It would shift along this line here. And you can do something similar for the shift between all trans retinal and all trans retinol as to how things might shift along the line here. And so with this, we now start adding in our actual data to compare to the phaser fingerprint. So in the upper left is that typical fluorescence intensity image. For these, we actually used larger fields of view than we had typically used before. And you can see the rod and cone mosaic. And here we see our ML cones, our rods, and our S cones. This is a phaser 4. Now in this case, each data point is a pixel in the intensity image. And what we noticed was that the rods tended to be closer here, very clustered around where the all-trans retinal was located. The ML cones then seem to have a larger proportion of all-trans retinol. And that was actually surprising to us. We actually were thinking that most of our fluorescence was coming from the retinol. It's thought to be the stronger of the two signals. But it looks, in this case, like we have quite a bit coming from retinal. The S cones are sitting out here, almost looking like they're shifting towards elastin, but I don't think there's elastin in S cones, just to be clear. I think there's a fluorophore. There's something there that we've missed. We need to find what's going to be out here with a longer fluorescence lifetime that's going to shift those S cones in that direction. And so we're looking now to what potential molecules we could consider to characterize in order to have as a possibility within our S cones. We color-coded the phaser fingerprint, or the phaser map here that you can see, and if we take that color code and then reapply it to our fluorescence image, then we have what we call a phaser image, which then shows the color coding. You can see here very clearly the delineation between the rods, the ML cones, and the S cones. So as we start to probe S cones a little bit further, we found that they changed with stimulation. And so this is a fluorescence intensity image, unremarkable from top to bottom. You don't see any differences. But in fact, we had imaged the top portion originally and then moved down to do a second image. And so the top half, shown in pink, is previously imaged, and the green portion of the bottom had not been imaged previously. And again, keeping in mind these are anesthetized, paralyzed primates, so we have very good stability of where we are with our image location. There's not a lot of motion during imaging. And so this is the phaser intensity image. And what we found was that the S cone sort of arm extended out much further in this direction. So now we are really aiming, is there a fluorophore or something out in this region that would pull it in this direction that we're interested in? And we did a little bit of k-means clustering to separate into two clusters, the sort of normal lifetime cones and the long lifetime cones. This time we've color-coded them in orange and blue. And when you look at the image, you can see very clearly that the previously exposed region tended to have this longer fluorescence lifetime, and the non-previously exposed region tended to have S cones that were within the normal range of what we'd expect. Again, we don't have a good answer yet as to exactly what's happening, but we have ideas and a guide to sort of lead us to, we need a long lifetime component, and what is that and what's happening, and what makes S cones different from the ML cones. The stimulation comes from our imaging laser in and of itself. It's monochromatic. That's a 730 nanometer stimulation. It's just the imaging beam of itself. It's so bright that it leads to stimulation. And interestingly, Christina Schwartz, when she was postdocing with us, did calculations, and the ML cones are actually much more stimulated by the 730 nanometer light than the S cones are. And so it's sort of an interesting quandary as to what's happening. Is it a two-photon vision phenomenon? What exactly is happening? And she's been looking at that, I think, in more detail as time progresses. We then moved from the peripheral regions where we were looking, where we have the nice large cones and the small rods, into a fovea. Unfortunately, we only do one image at each location. We don't go back and repeat the images just to ensure that there's no later effects. And so we get one shot in each monkey. So we have very little data in the fovea, but we're really excited to get this bit here. And so this is the first time we'd imaged this fovea, and you can see the intensity image, not as sharp as some of the others. I want to point out that in these cases, the fluorescence lifetime data is lower resolution than we are actually able to achieve with our typical AOSLO imaging. I'll come back to that as well. But what you can see is very clearly the rod peak has disappeared, and the whole cluster has shifted up towards the retinal. And so for some reason, these foveal cones almost seem to have more maybe retinol, more FAD, more bound NADH. Hard to say exactly what it is yet, but they're shifting up there. But you still see the S cones here. You see very clearly the S cones falling in this region here. There should be some S cones within the fovea as well. And so start to probe that as well with something we might be interesting to look at down the road. We looked then into the inner retina. What's happening there? We don't have the retinoids. We're not going to have the visual cycle taking place. So this is an image in the ganglion cell layer. It's a little more difficult to see. If you look in the upper left image, there's sort of rings in a sense. The ganglion cells tend to look like almost pearls on a string or little beads, and tends to have rings. This is not one of our best ganglion cell images, but again, the resolution here is lower than what we'd like to achieve. But we can get the fluorescence information, and we can see that it's a combination of various metabolites, NADH and FAD. It's a mix of those in that image. If we move into an area near the optic nerve head where we're looking at a branch retinal artery on the left and a branch retinal vein on the right, we can see a very clear delineation between the nerve fiber space and the vessel walls, particularly on the artery. And we can see that that is actually shifting towards a fluorescence coming from the elastin. And so what's interesting is, you know, when you start thinking about it, the intensity image gives you fluorescence is there or not there. But it doesn't give you the information of what you're actually looking at. Whereas when you start to look into this fluorescence lifetime in the phaser space, we can start thinking about, well, this is the fluorescence coming from the elastin, this is the walls, these are the elastin here. This is more like the ganglion cells. This is more nerve fiber layer, more neural retina. There's the information there, and start looking at how that shifts. The key questions will start to be, how does this change in disease processes? How does that shift when you have cells that are dying and unhealthy and the metabolism in those cells changes and the ratio between NADH and FAD shifts? That's the next step of where we want to go with this. Monkeys are very difficult to work with. We get few of them. There's not a lot of disease models in the monkeys. And so in moving to Waterloo, we're shifting all of this work to be done in mice so that we can start looking at mouse models of retinal degeneration, mouse models of outer retinal degeneration, glaucoma models, various things to look in the inner retina there. As we move forward, I sort of mentioned a couple times, the images are lower resolution than we would like. And there's a lot of challenges that we faced in doing the fluorescence imaging, the fluorescence lifetime imaging with our adaptive optic scanning light ophthalmoscope, particularly because of the speeds that we're imaging. A lot of the fluorescence lifetime techniques have been developed for use with microscopes where you can sit for a longer period of time on each pixel and dwell on each pixel, capture enough photons, and then move to your next pixel. We don't have that luxury because we're imaging at 25 hertz approximately. And so we have this not resonant scanner, so we have sinusoidal distortion. And so what we actually, the green square here shows you what we would capture on just the forward scan of our AOSLO images. And we'd be capturing those at 30 megahertz for the frame rate, for the pixel rate. So we have sinusoidal distortion. When we image with the fluorescence lifetime, we actually crop our images down to remove the sinusoidal distortion. So the images I've shown you up until now all have distortion within them. We've just cropped them down so that you're only seeing this small region. So we're throwing out a lot of data, a lot of information that we would normally have. You also lose information in the back scan. The laser is on continuously, so you have photons being excited, exciting things on both the forward scan and the backward scan. Typically, if we were just doing intensity-based imaging, we'd combine the forward and backward scan together. We, again, have not been able to do that until recently. And of course, as we move into humans and into mice, we want to consider that there's more eye motion. And so with the eye motion there, we have to be able to compensate for that. Typically, we would take the reflectance image, look at the motion in the reflectance image, and apply that to our intensity of the fluorescence images and create intensity images that have been shifted to compensate for the motion seen in reflectance. We were unable to do that until recently with the FLIM data. And finally, the Becker and Hickel boards that we were using for TCSPC, their pixel clock, their collection is much slower, so we had larger pixels than we would have liked, and so we did not have the same pixel size match between our AOSLO images and our AO-FLIO images. And so we want to correct all of these, and fortunately I was working with a great team of programmers and electronics engineers, Keith Parkins and John Yang, who developed software that we use now in post-processing, and we can go from, this is an example in primate, from a uncorrected, so this has no optical tracking. We took off the optical tracking. The images I've shown you previously all had optical tracking. This is uncorrected, straight collection. You can still see that there's some cones there. We captured 2.9 million photons. You can see the difference between rods and cones when I show you the tau mean image. But if we are able to correct the data, we can see very clearly now the ability to remove the sinusoiding, remove the motion, and actually do registration, and not quite, but getting close to doubling our photon count by combining in the backward scan as well. So we're now up to 5 million photons, and if any of you start doing photofluorescence lifetime, counting photons and the number of photons you get, the number of more photons the better, unless you get too many in the eye. We're never in the situation of having too many photons. And you can also see, though, very clear separation. These are much sharper. The sort of appearance of the cones looks much sharper than it even did in some of our other images. Very clear delineation there. So we're really pleased with this software. And so the next story I'm going to tell you, where we move into humans, really relies heavily on this new software that we've developed and these new advances that we have in our post-processing. And that's to delineate a person. I always like to say, that was my eldest son's self-portrait in kindergarten. And so I think it's a nice little picture of a person. So clinically, fluorescence lifetime has been used and shown several differences with age. And again, they're looking at the entire retina. There's not a lot of confocality here. But you can see longer fluorescence lifetimes with age, and I will just orient you. They use a different color scheme than I typically use. I use a microscopy-based color scheme because we're looking at a microscopy scale, in my opinion. So here, blue is long and red is short, opposite to what I've shown you. But you see increasing fluorescence lifetime with age, and we can see with a host of diseases how various fluorescence lifetime patterns appear. And so this is promising to start understanding it, but we were interested in what if you could isolate into just looking at the RPE layer. When they do their fluorescence lifetime, they have conflicting they have errors from the lens fluorescence, macular pigment plays a role, and they're looking at the entire retina all at once. And we said, well, what if we were able to target just into one layer? And so Charlie Granger actually started this project, and he had been, for his PhD thesis, doing imaging of the human RPE mosaic with fluorescence at 532 nanometers. And he was the first to start doing fluorescence lifetime. That was then taken over by another graduate student of mine. But beyond the intensity, we could use the fluorescence lifetime to try to understand more about what's happening, what changes with disease, what's going on in the retina. Are these the idea is, could we envision, are these RPE cells healthy or not healthy? Is there something that could tell us that we could look at? And can we understand what's happening with different diseases? And so Janet Tang took over from Charlie and has started to do the fluorescence lifetime imaging. And I showed you this earlier as a tau mean plot. But really, we wanted to move quickly into trying to understand the mechanisms of what the fluorescence origins were within this. And so she also did this phaser fingerprinting. Again, a host of solutions, the same set of solutions in her system to do human-based imaging. And we excite with 532 nanometers. We collect emission 575 to 750, so a very broad emission. And we're using this time a supercontinuum source that's been filtered. It has a pulse of 80 megahertz, and it's about a 50 picosecond pulse. So we've gone from about a 55 femtosecond pulse into photon now to a 50 picosecond pulse. And so we looked at 13 different people that Janet then compared here. So as we start to build our phaser fingerprint, we saw fluorescence from some fluorophores we didn't expect to see, but some that we did. A2E is a constituent of lipofuscin, down here, very short lifetime. All trans retinal showing up here. We have melanin fluorescence now within the human RPE, and we looked at FAD, and we added a whole host of other fluorophores in this spectral range. Collagen, different types, elastin, NADH, lutein and zeaxanthin, which were macular pigment, which were considerable problems for fluorescence imaging or are in the clinically fluo. They consider lutein and zeaxanthin to be fluorophores within that. So we didn't see any results from those. Those were all null, but we have this phaser fingerprint now. So a little bit less than what we could see with 2-photon fluorescence, but not surprising for single photon fluorescence. And again, here's our human data then placed on top of that, and we can see it's not purely A2E fluorescence. There seems to be some contribution from something else up here, and we actually think that the other major contributing fluorophore in this case is actually melanin. And as we start to look with age, this is each data point is now from a single location in a person at different eccentricities. We looked at 7 different locations in each person of 13 people. Here we're plotting it versus age. You can see in purple the younger eyes moving up to, in yellow, the older eyes at age 65. And what you see is this significant shift towards longer fluorescence lifetime in the older people. And this is actually consistent with what you would expect from an increase in melanolipofuscin with age. And this is also consistent with what we've seen in clinical fluo. And so we think that this is nice evidence in vivo of the melanolipofuscin increasing with age. Now, we were interested in trying to push this into disease, and working with Kristen Bowles-Johnson, who was a postdoctoral fellow with me. She and Janet imaged several different patients. Here's just a couple of examples. And an older person with pattern dystrophy in the middle, and in the right is a person with pentosam polysulfate sodium, retinal toxicity, PPS toxicity. This is a drug used for treating urinary problems, and it leads to a retinal toxicity that's very similar in its phenotype to AMD, but happens at a much younger age. And so Kristen's actually been using this as a model of AMD and looking at various characteristics. And these are just some sample images, reflectance on the top, fluorescence in the middle, the mean fluorescence lifetime in the bottom. We could see a shift towards longer fluorescence lifetime in gray in the PPS toxicity subjects. We're not sure if this is an age effect, because this was a normal looking location in the pattern dystrophy patient, or if it's at all related to disease. And we could see a clear shift in phaser space. We found in all of the PPS toxicity patients that Kristen's looked at, this shift that the clinically noticeable on OCT diseased areas had longer fluorescence lifetimes. The exact reason for that, we really want to probe. And then finally, as I wrap up, I just want to show you one new thing that we started doing. This is brand new. Janet literally finished this figure and sent it out in her thesis Saturday. So I stole it from there because I liked it and I wanted to share it. So Janet has now started taking the RPE images and outlining individual RPE cells. And if you overlay that onto the fluorescence lifetime image, you can look, if you look really carefully, if you look in every individual cell, there's sort of a variety of things happening in each cell. And we can start considering those. But we also said, well, what if we just looked at the average of a cell and said, okay, we're going to sum all of the pixels within that cell, generate one histogram, and create one lifetime for each individual cell. And if she does that, she actually sees a much tighter clustering of the fluorescence lifetimes. Now they actually also, this is a histogram of the fluorescence lifetimes. In pink is the pixel-based image on the left, sort of a histogram of the fraction of pixels per cells for the basic pixel by pixel analysis. And then in the darker red, she's showing the longer fluorescence lifetime where she actually did a lifetime for each cell. We start to see less variability in our normal healthy eyes, and the next exciting thing to look at will be what happens when we go to diseased eyes. The other interesting thing is in phaser space, it's a little hard to see with the colors here, but this, the cluster is very tiny. It's just that little tiny spot there. And so by doing this analysis, we're actually able to really tighten and narrow down what our fluorescence lifetime values are for that distribution of cells. Now, Two things to think about. This works really well when you are light starved because we can pool all the data within a single cell to create one data point. So that's sort of our motivation for doing that. And I don't have the data here, but as we start to move to other excitation wavelengths, other spectral emission collections and narrowing down emission bands, we start to lose photons. As we start to lose photons, it becomes very difficult to analyze in the way pixel-based that we were doing on the left. By doing this, we'll actually be able to expand the number of spectral properties that we can consider, the number of smaller spectral bands that we consider, and start looking, potentially delineating those into smaller regions. That's exciting to look at potentially sources of different fluorophores. The other thing that's interesting is just looking on a general global scale, how do those change? At the same time, when you have very severe disease, like I showed you in some of the previous things, this pixel by pixel analysis will be easier to do. We can't always outline cells. And so this is I think a great way to start looking at early disease changes, but will not necessarily be helpful for us as we go to late disease changes. So again, a combination of both methodologies is what we want to continue doing in the future, but it's exciting. I am excited to see that we can actually delineate this and start getting this pixel by pixel analysis converted into a cell by cell analysis. And so I hope that I've been able to convince you through the two examples that I showed you that fluorescence lifetime can add a whole new dimension to what we're looking at with fluorescence, and especially at the cellular scale, both in the 2-photon fluorescence imaging, such as the images on the left, or in our human-based imaging, and that it can help us start to delineate different functionalities and different functions between the cells, and hopefully we are moving that now into looking at retinal cell health. And finally, I will thank everyone who played a huge role in all of this. Couldn't have done it without all of these people: Kartik Kanala, Keith Parkins, Kristen Bowles-Johnson, John Yang, Janet Tang, and Kang Huynh, and of course our funding sources. Thank you. So we've been using an 80 megahertz laser in both cases. The phasor plots can be evaluated at the pulse repetition frequency, but they can also be evaluated at, other orders of the pulse repetition frequency. So you can evaluate and shift things within phasor space, yes.
And that's what's providing you the x-axis in this phasor plot, right?
The change in the pulse repetition frequency is not providing the x-axis. It's the sine and cosine components from the discrete Fourier transform. We evaluate it at one frequency. It's because it's one data point, right? You do the Fourier transform, you select one data point. You can select different orders and create different phasor plots, but you only get one for each repetition. For that repetition frequency, you only get 1 point for each. That's a very good question. So John, that's actually a very old paper from John now. He does it, I'm going to do a poor job of explaining it. I'll start with that. He looks at the match between the forward and the backward scan and has a prediction of what it should be, and he updates that in real time roughly every few seconds. To learn more about it, I really would encourage you to look up a publication by John Yang.
That's purely processing, right?
It's purely data processing.
I wonder if you use faster scanners, you can avoid this issue.
If I could get a faster scanner, I would. We've had difficulty finding anything that would go at the angles that we need at anything higher speeds than that. So this is a post-processing correction for the sinusoidal distortion. Elf Dubra's lab has actually generated a new method that they're using. I think they have a paper on that as well to do some more real-time based on recordings from the actual scanners, as opposed to ours is image-based correction, and they do a scanner-based correction.
Okay, thank you.
Okay. Thank you so much again for interrupting your vacation. That was a great talk. And I think maybe I didn't catch that, but could you comment a bit more on the age dependency of the human RPE mosaics?
Certainly. So, right, I have a picture right here. So, what we find, clinical fluorescence lifetime finds it, we find it as well. In longer fluorescence lifetimes across the retina as you age. So there's no interaction here with where we were imaging. The motivation for starting to look at this for us came from work out of Christine Curcio and Thomas Ock, who had shown that the molecules within RPE cells, the composition of RPE cells, changed, and it was differing as you aged, and it was differing with eccentricities. And so they have, I don't have the paper here, I wish I did have it. It's gorgeous figures where they showed that these, basically what you get within a cell is different. And so we think that that difference of relative contributions, and that's what is key here. We can't, I'm not going to tell you the absolute concentration of A2E versus melanin, but the relative concentration between things shifts things within fluorescence lifetime space, within the phasor space. And so what we think is happening is as it's shifting, as it's getting older and you're shifting towards the left, there's more and more contribution from melanin fluorescence than the A2E fluorescence. Now with the caveat that, a couple of caveats that, A2E is only one constituent of lipofuscin. So one of the things we'd like to add to this are more constituents of lipofuscin. Melanin, that's just one type of melanin, and it's a melanin we were able to purchase. It does not guarantee that it's the exactly same melanin that is in the RPE. So these are representative of where we think things are happening, but with that, we think it's the increase in melanin and the presence of melanin within that, and that's consistent with ex vivo results that have been done in histology.
Thank you.
So you said that with different pathologies, you saw a shift in the fluorescence lifetime, but that was for different pathologies, right? And my question is, it's always the same shift? Because for different pathologies, there were different. I mean, the fluorophores that change should be different, right?
Absolutely, absolutely. So what's the shift and where's it coming from? And that is the next direction. So we see the shift in the PPS toxicity subjects. If you look clinically at FLIO, they see some differences where they do get longer, they do get shorter in different diseases. We're looking to see, we haven't imaged a host of diseases yet. And so as we start to look at more diseases, we want to see, do they maybe shift not just in one direction to get longer, but you can get longer. You can get a longer fluorescence lifetime and go here, but you can also have a longer fluorescence lifetime and be here, and those would be different fluorophore contributions. And so what we need to actually start doing next, and this once we get the systems up and running in Waterloo, start broadening and start looking at more diseases, more potential things. And again, in the mouse models, when we look into photon fluorescence, very specifically targeted diseases with genetic mutations that we know what we should be expecting, and use that information to help guide us in interpreting data maybe in diseases that are not as well characterized.
Well, another question. There is all these papers using the Heidelberg system, the clinical one, which basically measures lipofuscin, and they're able to measure differences between central and peripheral retina and some other things. And I remember talking to Janet, like there are some data that are different from those obtained with the clinical one and your higher resolution system, I guess. But then she said that because you had this higher resolution, well, I didn't understood very well what she said, so.
All right, so you're talking about the data that I left out.
Okay, sorry.
No, so we have, we're very fortunate to have a collaboration with Paul Bernstein, who has a clinical Fleo system. So we have taken some of our people, and they've been very generous and flown them down to Utah, and they've been imaged on the clinical system down there. And we've been able to compare now between our clinical Fleo system analysis there and the adaptive optics Fleo that we've been doing. The reason I didn't show the results is because we haven't actually met with our collaborators yet to tell Paul what results we've found. Admittedly, they're in Janet's thesis at this point. But we're meeting with him actually when I get back next week. Long story short, without letting the entire thing out of the bag before we've had a chance to tell Paul about it, we see some similarities between the two systems, and we see some differences between the two systems. And some of those similarities and differences will we believe stem from our ability to isolate the RPE layer versus their sectioning. But surprisingly, we see fairly good correlations between the two, which sort of surprised me in some ways, not in others. It's depending on how you look at it.
Because the clinical one, I don't know which, they don't differentiate between layers, right? It's just a general...
The clinical one's going to have the entire retina, plus they have an artifact from lens fluorescence.
Ah, yeah.
And so there's minimal confocality there. And so they have the entire thing. So one of the things that it makes it really interesting is, and that we haven't done yet, is they're getting fluorescence from the entire thing. We don't match perfectly. So there's fluorescence coming from somewhere else. I'm really interested in looking at next, is there fluorescence coming from the inner retina? There should be FAD out there. There should be a lot of fluorophores up there. Can we actually focus and image a fluorescence information in the ganglion cell layer, for example? This is the direction we're heading. It's something we haven't fully tried yet. But again, we're light-starved as it is, so going up to the inner retina for fluorescence, we're going to be severely light-starved. And we're going to have to be very careful about the any fluorescence, residual fluorescence from the RPE.
Again, thank you for coming to visit us on your vacation. That's really nice of you. Yeah, we'll make Maria buy you lunch later. It was a great talk, and I'm really curious with there's so much information you have to get with multiple fluorophores in the eye, and having the lifetimes change so much with the being bound, being free, maybe if there's a different concentration of things in there. I'm curious if the concentrations are important. It's a two-part question, I guess. If the concentrations are important for what you're looking at, pixel by pixel or on average, and do you use any sort of like maximum likelihood or least squares to kind of deconvolve what the different concentrations are doing in the image?
It's a difficult question to answer. It's a good question.
Sorry about that.
No, that's good. We are very conscious that absolute concentrations can play a role. So we're very interested then in relative concentrations. And so we often have these discussions and debates. I think of it as your absolute concentrations dictate your fluorescence intensity. Your relative concentrations dictate where you are in phasor space. And so differentiating those becomes important. We've done some analysis to look at, does the location in phasor space correlate with the intensity of the cell? It doesn't. We haven't found any of those correlations that were strong at this point. So bright cells, lots of fluorescence doesn't necessarily mean that you're going to have a particular space, a particular location, in a particular lifetime. Does it matter? Yes. If you have, and I'll give you an example of data I didn't show. We have done some work with extrinsic fluorophores, adding fluorophores in that have a lifetime variability and trying to measure those. We put them in RPE cells. RPE cells are very fluorescent with a lot of lipofuscin. And the expression of the fluorescence that we put in is variable. And so that background lipofuscin fluorescence can really overshadow, in some cases, the expression of the cell and the fluorescence that you're actually trying to measure. And so concentrations of fluorophores can, depending on what your target is and what your goal is, can really play a role and can confound what you're trying to look at. And so then you have to start getting creative about how you're going to analyze the data and how you're going to pull out the backgrounds, and we haven't solved that problem yet, hence that paper's not published yet. But we're working on it, and we have a lot of ideas about how we're going to do that. But I think you just have to be conscious of those factors as you're starting to look at this. It's not, this is not a slam dunk. This is not a, here's a data point and this is definitely this combination. As you start having multiple fluorophores, you can start to see it could be within a triangle, it could be within 4 different fluorophores. It's starting to think about what the potential contributions are, and then we have not done any maximum likelihood estimations or anything on the phaser space to see, is it this, this, and this. We haven't done that, but there are thoughts of doing that. There's actually a lot of groups starting to think about deep learning ways to combine things, so that's something we might think about in the future.
Thank you for your talk. I was wondering if the fluorophores that you are measuring in the anesthetized monkeys behave the same as if the monkey was not anesthetized, and if you have to take that into account when doing the fingerprint.
That is an excellent point. It's one that we often overlook, but yes, the anesthesia will change the metabolism. It will change how things are functioning. Unfortunately, we never were able to work in awake monkeys. Be great if you could down the road. But yeah, it can be a factor. It can slow things down.
Thank you.
Yeah, let's thank the speaker again, and thank you for coming.